Terima kasih Dea. Pertama-tama saya ucapkan terima kasih untuk semua yang sudah hadir, juga Dea yang sudah memoderasi, dan juga tentunya untuk Sebastian. Terima kasih banyak sudah bersedia dan meluangkan waktunya untuk sharing di sesi skor hari ini. Skor hari ini merupakan season kedua setelah kita lakukan yang season pertama tahun lalu, tahun ini dari mulai Desember hingga sekitar bulan Mei atau Juni, di mana kita ada 17 sesi presentasi tentang restorasi terumbu karang dengan beragam permasalahan dan lika-likunya. Dan sesekali kita juga mengundang partisipan atau mengundang saintis dari luar untuk sharing ya, terutama dari sisi sains. Jadi itu yang kami harapkan gitu ya, untuk belajar banyak hari ini dari penelitian Sebastian sebelumnya, dan saya I believe this was from your research in Bunaken ya Sebastian. Ya, from Sebastian's research in Bunaken gitu ya. Jadi ini bisa menjadi suatu pembelajaran. We can learn so much from this, that restoration is not only like growing or extend the coral coverage or increase the number of fish, but also trying to see it from the scientific perspective, from different angle, and then how actually you can write or publish a scientific paper based on the work on your restoration, and of course publishing your work, it will be beneficial for many people who want to improve their restoration practice in Indonesia. Saya bersyukur sekali ya, dengan adanya skor ini kita udah lumayan banyak connection or network of reef restoration practitioner di seluruh Indonesia gitu ya. Semoga tahun depan kita bisa bertemu offline atau mengadakan seminar atau simposium atau workshop bersama-sama dengan para restoration practitioner. Kira-kira tadi Kepala Departemen yang harusnya memberikan sambutan, Bang Cacu, juga berkalangan hadir karena ada undangan lain. Bang Begin mungkin agak sedikit terlambat, tapi sepertinya kita mulai saja acara ini. Saya persilahkan kembali. Terima kasih Sebastian ketersediaannya, dan Dea saya kembalikan untuk memoderasi skors kita kali ini. Terima kasih Dea.
Thank you very much, Dea, and thanks also to Dr. Trias for the kind invitation. It's a great honor to be presenting in the SCORES seminar. I was watching the SCORES seminar with great interest from afar, although I didn't have the privilege of joining so far. So it's a great honor to be invited and to be able to present in front of this network. And my apologies if I give the presentation in Bahasa Inggris and not in Indonesian. But if there are questions afterwards, I'm also happy to lead the discussion or to respond to questions in Bahasa. Yeah? Just to check real quick, are you seeing the presentation mode or are you seeing the correct presentation, the full screen?
Yeah, it's
full screen. Okay.
Full screen, all good.
All right. Yeah, so I was really honored when Trias had asked me to give the presentation, but at short notice, the one thing that I could present is the work from my doctoral thesis, which hasn't been much published. So several of the things have not really been properly published yet, and so that's why it's good to be able to talk about this. And also, if things are published in a PhD thesis, not a lot of people have access to it, particularly if it's sitting on a shelf here in Germany. So it's good to be able to present my findings again to you, being mindful that this whole study is about 15 years old already, so keep that in mind. But still, until today, there are very little scientific ecological studies of the scientific aspects or ecological aspects of reef restoration. So as Trias has said, there is the practice of reef restoration, and we can learn a lot from the practice, but there is also restoration ecology, which is more driven by fundamental research interests and fundamental questions that can inform reef restoration practice. Yeah? And my study is one of those where I looked at specific aspects of reef restoration. So these two things should not be confused, but until today, there are very few studies that actually look at, for example, how fish communities respond to reef restoration. And so I think the results are still valuable, and this the same goes for coral recruitment. There is very little study of how coral transplantation actually affects recruitment. That was a burning question 15 years ago, and it still remains a burning question nowadays, I would say. So fish community and coral recruitment responses to reef restoration. You see here an image from us when we try to build up the experimental sites. This is in North Sulawesi, and you will see more of that in the presentation. Okay, it's moving. So what am I going to talk about? Before I go into the study itself, I'm going to talk a little bit background reef restoration. What is it? What are concepts and techniques, and why do we do it? Before then going into the study itself. So the study approach, what are the questions that we were asking, and what kind of methods did we use? And then of course, talking a little bit about the results, and then finally wrapping this up with implications for restoration practice and some outlook on some additional interesting aspects. So first of all, coral reefs and reef restoration. I'm probably preaching to the converted here. Reefs are super valuable ecosystems, particularly for a country like Indonesia, where 20% of the world's coral reefs are located. Reefs are the most valuable ecosystem in terms of area. So just from tourism alone, there's been an estimation by Mark Spalding and colleagues that $40 billion per year are generated by tourism, and we can see that exemplified in this picture here, where you have land-based tourism that is attracted to reefs. You have snorkelers, you have water sports people, you have people that are looking at sharks, which of course occur in healthy reefs. And so there's multiple benefits that reefs generate just in terms of tourism and fisheries, but also in terms of coastal protection. Almost 100% of the incoming wave energy can be deflected by healthy, structurally intact coral reefs. And once the reef is degraded, then this service provision is reduced. Reefs are, of course, also highly at risk. We all know this, particularly because of local anthropogenic factors. Climate change is increasing and is putting additional stressors to this. But at the moment, local anthropogenic human-induced factors are still the main cause of reef degradation. We have, for example, destructive fisheries. We have land-based sources of pollution. We have habitat degradation from construction activities. And if we're looking particularly at Southeast Asia, the Coral Triangle, Sulawesi, the Sunda Islands, and the Philippines, that's where there's a lot of local stressors on reefs. And so my three main points here that I want to emphasize: reefs are an important resource for livelihoods and for the economy. Reefs are threatened, and reefs, when they are degraded, recover very slowly. This is an image from Bunaken, actually, from one of the experimental sites, where there's a lot of rubble on the slope, and this partly resulted from net fishing activities in the area before the national park was erected. And there are estimations, if a reef is degraded like this, that's something that Dr. Trias is also actively working on, looking at rubble and rubble recovery. It can take a very, very long time for these reefs to recover. More than 100 years are some estimations, depending on the environmental conditions. And so this means we cannot only rely on natural recovery, but we need to assist the recovery with human intervention to help reefs recover. And not to create new reefs, that's not possible, but to intervene and to assist the recovery of reefs. Okay, so what can we do? There are some important concepts for restoration. So first of all, restoration itself is the recreation of habitat, very similar to what it looked like before. So this could be in terms of the function, species composition, the biological diversity, or the structure. And we're trying to get back to what the reef looked like before as much as possible. With reefs, of course, that is a problem because reefs are at the moment changing because of climate change, environmental change, and so on. And so to go back to the old configuration is sometimes difficult. That's why we're also trying something that's slightly different. That's called rehabilitation, and this is trying not to get back 100% to what we had before, but to focus on, for example, particular functions and qualities, or to replace them with an alternative set of species, assisting natural recovery. And the community in the end may not necessarily be the same as the previous one, but it's fulfilling similar functions. And so this is applying the idea of resilience thinking, or of functional ecology, where we're looking at particular processes that are valuable to nature and to humans, and we focus on these processes rather than trying to restore species that existed before, but maybe don't have a future in climate change. So it's a little bit more adaptive and forward-looking. And then finally, we can have the creation of completely new communities in areas that did not previously have coral growth. So for example, this can be done by coral transplantation, or by modifying the environmental conditions in a way to allow coral survival and growth in areas where there was no reef before. Oftentimes this is used when it comes to artificial habitat creation. But I always caution, carefully look at the environmental conditions, because if there were no reefs before, usually there is a reason for it. And it's difficult to create functioning reefs in a place where there were no reefs before. But just to mention that creation is also something that sometimes is done. Now, coral reefs, of course, are biologically complex habitats with a rich structural complexity and biological diversity. Here's an image from the Great Barrier Reef, and that's what a healthy reef looked like. So very diverse and structurally complex. And they're, of course, home to lots of other organisms that are reef associated, in particular fishes. Fishes and coral reefs in the Indo-Pacific region are almost 5,000 different species, and the coral triangle itself nearly 3,000 species. And this includes several groups that have important ecological functions, such as herbivores, carnivores, planktivores, and corallivores. And these were the species that I looked at in more detail in terms of community composition in my study, and we will see some results for these particular groups later. Now, with habitat degradation, we're losing structural complexity, and we're also losing living organisms. So we have two things that are being lost when a reef is degraded, and sometimes it's difficult to specify the relative importance of both of these. Are we losing organisms because there are no more structures, or are we losing organisms because the living coral is gone? And so that was one of the motivations for this particular study, to look at the relative effect of living substrate and of structural complexity. Now, coral reef restoration is a relatively young discipline. It developed over the last 3 to 4 decades. A lot of the concepts are borrowed from terrestrial restoration, where many methods were originally developed and then applied in the marine realm, for example, for mangrove restoration, but also for reef restoration. And there is an increasing number of books and manuals. These were the ones when I did my thesis, but since then, particularly in the last 3 to 4 years, a whole range of new manuals and instructions have been developed, for example, by NOAA, by the Americans, and from Australia, and UNEP in the frame of the current decade for ecological restoration. So a lot of development, particularly at the moment in this. And importantly, reef restoration includes several concepts and approaches. We already talked about rehabilitation, restoration, and design or creation. But there are also, you can also differentiate from more passive approaches, such as the implementation of marine protected areas, DPL, Taman Nacional, these kind of measures, to active restoration. And active means we intervene or we change something in the environment, such as consolidating the substrate if it has become loose, so to make sure that the rubble is fixed, removing alien material. For example, if there has been a storm, we take out debris, or it could be construction of artificial habitats, and even coral transplantation. And it's this active restoration that is the focus of this study. Okay, so before looking at the questions that I was asking, just to look, have one final look at some restoration methods. I mentioned artificial structures and coral transplantation, and particularly in the Indonesian context. And again, Dr. Theas has a lot of experience in this because she's surveying these. There are several main methods that are widespread, but at the moment, or by now, I would say hundreds of different methods and materials have been applied in the Indonesian context. Not all of them have been thoroughly studied. This is the example of reef balls, which were deployed in many places, particularly North Sulawesi and Sumbawa. These are eco reefs, which have been deployed in Bunaken, and which Trias is monitoring for nearly 20 years. But there is also coral transplantation. This is one of the students who was working with me from UNHAS, Onal, and Onal is gluing coral fragments onto little bases here, and we use these little bases and individual corals to attach them in the reef and then monitor them. You can also, rather than fragmenting corals, transplant entire colonies, such as we see here, so for massive corals, or you could even farm coral species. And that was an inspiration for the study. This is an image from Karimun, Java, in central Java, a coral farm there, and they are growing corals for the ornamental trade on these substrates. But you could, of course, also use them for reef restoration as a source of fragments without having to damage mother colonies. If everything is regrown, then you don't have a negative impact on the reef. And usually these things are grown on culture substrates, as we see here. If you can take these entire substrates and put them in the reef and they continue to grow, perhaps you don't even need to attach them further. And so this would take away one major source of work, of labor, if you can use this for reef restoration. Now, some potential effects that one should look at when studying reef restoration. From structures, we can have a substrate for coral, so new coral recruitment, and also fish habitat, so fish that are attracted to the restored area. Coral transplants, of course, also can generate fish habitat and are either a source of larvae, so they can produce new larvae that settle in the vicinity, or they can also produce chemical cues and so serve as attraction for coral recruitment. So coral transplantation can lead to an increase in coral recruitment in two ways: direct reproduction or attraction. A source of larvae or an attraction of larvae. And that's the background for my study. I wanted to look at fish community effects, so effects of structures on abundance and composition of the fish community, and the effect of the coral transplants on the fish community, as well as looking at the effect of location. So where you do the reef restoration, you have the natural reef environment. How is that affecting the community, fish community development? And then I looked at the coral aspect, so coral ecology, effect of transplants on coral recruitment. And lastly, I was interested, can we use corals that are grown on substrates in farming operation, put them into the reef, and how successful is this for the coral? So what's the survival of the transplants on this bamboo substrate? We were using bamboo because that's easily available, it's easy to produce, and it was also used in the farming enterprise that I worked with in Karimun Jawa. Specific hypotheses to look at: coral transplant and artificial structures lead to significant increase in fish abundance and species richness. That's a univariate response. Fish community composition differs in terms of species composition and relative abundance between untreated rubble plots, so that's my control, plots with artificial structures, and plots with structures and coral. That's a multivariate question. There are significant differences in fish community composition among locations with different environmental conditions. So the location matters. Again, that's a multivariate test. And lastly, coral transplantation significantly enhances recruitment and alters the composition, the family composition of coral recruits where there are transplants present. And that's, again, a univariate question. Okay, where did I do this? 3 locations in North Sulawesi. If you're not so familiar with North Sulawesi, here's a more detailed map. If we look at the tip of North Sulawesi, Ganga Island is this little island here, where I worked at a shallower site on the reef slope up to 10 meters, and then 2 locations in Bunaken Island and Teluk Manado, close to the village of Meras. That was a deeper site. Just to give you an impression, so this was Ganga Island, Meras in Teluk Manado, and the reef slope, Zona Inti of, or near the Zona Inti of Bunaken Island National Park. At each of these sites, we had 3 treatments: a control treatment, that was just the rubble plot, where we only put in markings on the side so I could recognize the plot and monitor the fish community. And we had recruitment plates there. Then we had plots where we erected structures from concrete blocks, and we transplanted corals, and we finally had plots with only structures without any transplantation. And we used spawning and brooding corals because A, they were readily available, and B, because we hypothesized that brooding corals which release larvae that are ready to settle would perhaps settle close by. And so you see more of a recruitment signal when you transplant brooding corals than if you transplant broadcast spawning corals. So these are the 4 different species of corals that were used. 3 Acropora and 1 Bacillopora species. Now here's a blow up what these plots looked like. I used hollow bricks from concrete, piled them, 5 of them up on top of each other to generate these artificial structures. And then there was transplantation of 50 coral fragments in between, 50 per square meter. Monitoring, we did a fish community monthly visual censuses with 3 repeated counts. Coral recruitment we looked at with the settlement plates that were collected every 3 months. And we also looked at reference values from the ambient reef in terms of community composition and fish community. So coral community composition, fish community. Okay, now to some of the results. For fish community, Ganga, I'm just showing some selected results because these are the clearest. For Ganga, we did see that fish abundance increased almost immediately and continuously until the end of the monitoring period. So this is over a duration of 2 years, and we don't really see a leveling off. So there was an increase that probably would have continued even further in abundance. And here we see the natural reference sites. So in the end, in the transplantation area, the fish abundance was higher than even in the natural reefs in the environment. And that's because the natural reefs in Ganga were quite degraded already. So the habitat that we created with the experimental plots was even more attractive to the fish community than the natural reef in the environment. And similar for number of species, here's the number of species. And again, we see the highest number of species found where we had corals and structures. It's beginning to level off a little bit. So the maximum amount of species probably is reached here. But mind you that we didn't look at pelagic fish, we didn't look at cryptic fish. So of course, this is only a small fraction of the fish community. But you see a strong relative and significant relative difference. Values at Bunaken were not quite so pronounced. So on Bunaken, we didn't really see much of a difference between the structure plot and the coral transplant plot. So there was a little bit more fish in the areas where we had transplanted corals. But if you compare it to the natural reef on Bunaken, the experimental plots were nowhere near as high as the natural plots. And the same if we look at the number of species, we have a much higher number of species in the natural than in the structure and the transplant plots. And this indicates the reefs in the vicinity were quite healthy, and actually there wasn't much need for reef restoration because the natural reef is much healthier than anything you could recreate. It's good, of course, to cover the rubble, but the fish prefer to stay in the natural habitat and don't go into the experimental plots. And this is very different in Bunaken, where the natural reef already was quite depauperate. We see this difference, or this lack of difference between the coral transplant and the structure plot here in Bunaken. If we look at the species rank plot, never mind the details, it's looking at the biomass distribution across the different species. And usually these things form slopes, as you can see here. My main point is to compare the different treatments. And you see there's really an increase from the control treatment to the structure treatment to the coral treatment on Ganga. And on Bunaken, it's really the structures that make a difference, not the coral transplants, because the reef in the environment is quite healthy. To look at what were the main drivers of the fish community composition. So what was the fish community at these different sites mostly influenced by? In Ganga, the natural reef had a hard coral cover of less than 5%. There were lots of soft and leather corals. And so these two factors were most important in shaping the fish community. In Maras, we saw that there was up to 1/3 coral cover, so slightly higher. But there was also crustose coralline algae as an important factor. So that's an indication of quite a healthy reef. And on Bunaken, again, CCA cover not quite so high, but a higher hard coral cover in the natural reef. And so coral cover was a strong driver of the fish community composition. And because at Ganga it was so low, it explains why the fish respond so strongly to coral transplantation. Yeah, so fish community correlated with depth as hard corals and leather corals and coralline algae. Those were the main factors. Now, if we look at multivariate fish community composition, this plot shows you the different treatments by symbols. So the triangles are the controls, the squares are the structured treatments, and those circles here are the ones with coral transplant and structures, and the colors show the differences between sites. We also see that Ganga is quite different in terms of fish community composition, and the Bunaken and Maras are quite similar, and particularly the coral transplant and the structures plots are quite similar. And we also see that the control plots are really dispersed. So the community composition in those control plots in a degraded reef is very variable. If you go from one degraded plot to another degraded plot, it's almost random what kind of species show up there or can be observed. And you get a more reliable and consistent community composition, as you see here, when you have corals present. That's when the different surveys yielded the same species. So we have a stable community of fish that is establishing over time, whereas in the control plots, each of these points is one sampling time over 2 years. We see that there's a lot of variability. Every time I came back, there were different species, essentially. So we don't have a stable community that's establishing. And it's just the statistical results from that. So there's a significant effect of treatment and of location, significant differences. Here I just want to show, maybe focus on the values on the far right. This is called relative dispersion. So that is looking at how similar or dissimilar the repeated observation or counting of the fish community was. And if you have a higher value, it essentially says there were more differences in the community. So I count again, I see completely different species. If it's a low number, then the community stays stable and consistent. And again, we see that in the plots where there's coral transplantation, we always have the lowest value, which means that the community is similar, stays the same. Whereas in the degraded reef, we see more variability. So variability is an indication for environmental degradation. Now, I said I wanted to look at some important functional groups, so fish that have an important role in the reef. Here we look at the abundance of herbivores. Again, it's a multivariate plot, so each of these letters that you see is one sample, one fish community sample. The letters indicate control, so there's a lot of dispersion here in the control values and very similar structures and coral. So the coral transplant plots are very similar, but you also see these little bubbles, and the bubbles show how abundant the fish in this particular group were. So we see that herbivores, so the species that feed on algae, are most abundant, of course, where there is coral, but they're also quite abundant in the plots with the structures. So structures are important to attract herbivores because algae grow on the structures, and that attracts the herbivores. But if we have coral, it means that there is an additional increase to attract herbivores. And if we look at different groups of fish, we see slightly different signals. So for those fish that eat other fish, the carnivores, they're most abundant where there is corals, but some of them also occur in the structure plots. If we have those fish that feed on coral, that depend on coral, the corallivores, they pretty much only occur in the coral transplant plots, and that's not a surprise. It means they are an indicator species for the presence of coral. And planktivores, the species like little damselfish that feed high up in the water column and transfer energy from the pelagic, from the open water into the reef, an important function to support the health of the reef. Again, these are mostly associated with having coral. So there is an important role of coral in the reef for reef ecology. In terms of the community composition, how similar they were, we see a couple of trends. So the natural reefs, I'm just going to go through this. The natural reefs in Bunaken and Meras were actually quite similar. they form one cluster. We see that the structures and corals and the structure plots, so the ones with artificial reefs and with coral transplantation, they're actually quite similar in Bunaken and Meraz. So again, coral transplantation doesn't make much of a difference. At the control reef, so these are the rubble areas, are also very distinct. And then Ganga, lastly, forms a cluster of its own. So very distinct fish community in Ganga, where the reefs are a little bit are different. There's a lot of soft corals, and the reefs are more degraded. Okay, moving on to coral recruitment. These are recruitment trends over time. And we see that the highest number of recruits was observed in Bunaken. There are some spawning peaks, so there's seasonality. Once in a year, there is an increase, so that's like a natural variability. And of course, that is because once a year we have mass spawning of species. That's reflected here. And if we look at the average numbers of recruits in the different plots, I'm just showing the results for Meraz and Bunaken. Keep in mind that Meraz, the scale is different. So the highest number was in Bunaken, but we see that there is no clear trend. At the final measurement, we did see an effect of the coral transplantation, but in most of the other treatments, there wasn't really a clear effect of transplantation. So this is the only significant values. We do see some significant differences, but in Bunaken, for example, here the highest numbers were not where we had transplanted coral. Only in the final observation period did we see a difference. This may show that there is some effect of the coral transplants after a long time, after 2 years, but it's a small effect. Now, what about species composition? We also looked at what kind of species are recruiting. Again, remember that I transplanted Acropora and Pocillopora species, and that's why I have marked them here. The results are always shown for control. Then the coral transplant and the structure plots is the last one. And again, you see there's pretty much no real difference in terms of absolute number between the treatments, with the exception of the final measurement, where the highest number was really where we transplanted coral. Again, keeping in mind that there are differences in scale. But if we look at the relative, so out of 100%, the relative composition, we do see a higher number of Pocilloporids, so the brooding species where we had transplanted coral. So maybe there is a small effect there of coral transplantation. Yeah, if we look at the relative composition and significant effects of this, we do have significant effects of treatment on the composition, and most of the Acroporas actually occurred in the structure and in the transplant plots. So maybe Acropora species are attracting other larvae from outside. That could be an explanation for this, because the species that we transplanted, at least here, are broadcast spawning. On Bunaken, we used the brooding Acropora species. So maybe these did release larvae and these were recruiting on the plots. So there's a small indication, but it's not really overly significant, or not a very clear signal. The last thing I want to show is the survival of the corals. On Bunaken, we had the lowest mortality over time. So by the end of the observation period, we still had less than 20% mortality. For the other coral species that we used, and particularly in Ganga, but also in Meraz, we had quite high mortality at the end of the monitoring period, and this also is related to disintegration of the substrate. If you remember, we attached those corals to bamboo boards, and here we see the degradation of these bamboo boards over time. By the end of the experiment, all of the bamboo had disappeared, had disintegrated, fallen apart, and at that time we see very high mortality of the transplants. So this is not a suitable method for coral transplantation. You need to make sure if you transplant corals that they are firmly attached for a long time, because otherwise you will see high mortality at the end. And it also shows that sometimes you need to monitor for a very long time, because the mortality only occurs after 2 or 3 years sometimes. And if you stop monitoring after a month or after 2 months, you may think, oh, it's all good, we have very high survival. But then actually the survival can drop dramatically after a longer time. So that's important to keep in mind. And the species that we had on Bunaken was this sturdy species. So this seemed to be, Acropora brueggemani seemed to be suitable for dealing with the environmental conditions and for dealing with the degradation of the substrate. So maybe that's a suitable species also for attracting larvae, but the other species we used were not doing very well. Okay, I want to revisit my hypotheses, treatment effects on fish abundance and species richness. We didn't see a clear effect of that. We did see a strong response in Ganga, but not at Bunaken and Meras. There didn't make a difference if we have coral transplants in addition to artificial reefs. We did see treatment effects on the composition of the fish community. And we had a strong effect of location on fish community composition. Yeah? So the fish community was significantly different between Ganga and the reefs in Bunaken and Teluk Manado. And lastly, with regards to coral recruitment, treatment effects on recruit numbers and the relative abundance, there were no clear, no consistent results from this. So further studies are needed. What kind of species maybe have an effect on coral recruitment? But my study was not able to confirm that you can increase coral recruitment by coral transplantation. To sum up, the effects of treatment for fish differed among sites. The effects were stronger in a depauperate area, in a degraded area, Ganga. Yeah, we had a stronger effect of coral transplantation, and transplantation overall had little effect when it was close to natural coral, as we saw particularly in Bunaken. If you have a high natural coral cover, you may not need to transplant coral. Artificial reefs thus should be used more frequently. Reconsider transplantation. You may not need to transplant coral, and you need to know the conditions at the restoration site. So this is really important in choosing the right method for reef restoration. Now for the coral, no consistent effect on recruitment, low survival of most of the transplants that we had, and very high natural recruitment also in the control plots. So again, if you have good natural conditions and high natural recruitment, you may not have to transplant corals. And that means artificial substrates may be more efficient than transplantation. Transplantation is labor intensive, it's very costly, and it may not be necessary. Just having suitable substrate, particularly in reefs in Indonesia, where the natural conditions are still usually quite good, and if you manage them appropriately, artificial reefs may be sufficient. And if you have to transplant corals, there may be reasons for doing that. Then make sure that they are well attached rather than using the method that I used. Some further thoughts. How are we doing on time, Trias? Can I still go on for another 5 minutes?
Yes, all good, Sebastian.
Great. Okay, thanks. So some further considerations. Can we define a benchmark to think of how much restoration do we need? So if we look at the environment, what tells us how much we need to restore? Do we just need artificial reefs, or do we need transplantation? What are the population dynamics in relation to treatments? So do we see a lot of fish migration to the plots, or do we see actual production, like new fish that are spawning and growing? Same for the corals. Do we see new production of corals, or is it more attraction? and what are the stabilizing properties of the substrate with regards to fish community. If you remember, the degraded reefs were very variable. There was a lot of variability, and we see very stable communities developing in the restored reefs. So what is it about restoration that leads to more stable communities? And lastly, if we see planktivores attracted, what are secondary effects in terms of trophic coupling between live coral and the fish community? So is live coral important for the energy balance and energy transfer? Because it attracts planktivores and because fish are feeding on the coral. So these are still some unresolved questions. And lastly, what is the effect of fishes on coral recruitment? Yeah, if we have more fish, particularly planktivores, they may also feed on coral larvae that are drifting in. So there may be a trade-off between having a lot of planktivores and having coral recruitment. These are still unresolved questions for students at IPB to address in the future. Okay. Just wanted to give two additional thoughts that I didn't present with my thesis, but that might be interesting. So the first thing is, I'm just going to click through this. If we look at immigration and emigration of fish, so we count at 2 time points, and we look at what kind of species are present. So we see that the first row is the first time where we count, and we see there's these species A, B, C, D, and so on present. We have a total of 6 species. In the second time when we come back, we have 5 species, for example, but they're not necessarily the same. Some species are new, some species have disappeared. And so by looking at the amount of new species, that disappear compared to the next time, that's relative extinction. If we look at those species that are new, compared to the total number of species, we have relative immigration. If we look at these 2 trends, we can do that for the different treatments, and we see sort of an equilibrium. Where is the amount of new species that are arriving compared to those species that are going extinct? Where is it similar? And that's sort of a carrying capacity that we can define. And if we look at the point where this carrying capacity is, so where extinction is the same as immigration, we have an equilibrium number of species. For the control, and here I'm showing that for the different control plots, so degraded, that's somewhere near 24. If we look at the plot with the artificial structures, that is somewhere 44, so almost double the amount of species carrying capacity. And then lastly, where we have corals, it's above 60. So we see a much higher capacity of the restored reef to contain species. And this could be used as one benchmark for defining restoration efforts. Because if we look at how many species, how many fish are surviving, this is influenced by the quality of the habitat. If we look at on the left-hand side, we have numbers of recruits, of fish recruits that are coming in. Not all of these will survive to maturity, which we see on the right-hand side. In a degraded reef, the carrying capacity, as we have seen, is lower. So here we have a recruitment limited community, so less fish are arriving than the community, the habitat can support. And so in this case, if we are below the carrying capacity and we have a low number of recruitment, then it's recruitment limited. If we have species that are arriving or larvae that are arriving above this carrying capacity, then only as many species and larvae will survive as the carrying capacity. Now, if we can artificially increase this carrying capacity, we push this line higher, then we can support more of the incoming recruits. And this could be one benchmark for restoration. We need to know what is the natural recruitment rate, and we need to know the carrying capacity of the system. And then we can use these two to inform the amount of restoration that is necessary. But the next one, I'm not going to go in detail because we're short on time. And I would like to thank you for your attention. And if anything, if I was talking too fast or the English was difficult, then mohon maaf ya, we can talk about it in Bahasa. Thank you for your attention.
Okay, Sebastian, I think it's very interesting information and important, especially for the practitioner and also policymaker to decide where and how to make an effective restoration, and also for the researcher to continue and maybe get the baseline information about the fish community and restoration. Maybe before continue to the question session, saya punya cerita atau sharing tentang pengalaman. Ini terkait dengan workshop yang saya lakukan last month. So I have 1 month workshop interdisciplinarity. It's from biologists and then ecologists, sociologists, economists, and government, and we talk about the MPA and sustainability. Jadi ada pertanyaan dari seorang ekonomis mempertanyakan bahwa sebenarnya kenapa dalam konservasi ataupun restorasi yang menjadi salah satu indikator yang diteliti atau dilihat itu adalah ikan. Memang harus selalu ikan ya? Gitu. Jadi itu salah satu pertanyaan dari seorang ekonomis. Kenapa ikan bisa menjadi indikator yang tidak pernah lepas dari konservasi? Jadi pertanyaannya, bisa tidak mungkin another indicators like carbon or nutrient? Dan di tim kita coba brainstorming beberapa menit dan kita juga bertanya, iya, kenapa selalu ikan ya? Seperti itu. Dan ternyata ketika kita berbicara dari semua aspek, for example, for me, from the ecologist and biologist, I think when I want to see the condition of coral reef, I also have to see the fish community. And then when people talk from this, from the fisherman side, for example, they want to talk about the economic or social activities, they will ask what kind of fishes they catch and at what, how many kilograms per day. That's important question and important information. And also from the economists, they also will talk about how many rupiah fishermen get per days, and that's also relate to fish. That's why I agree with Sebastian that here fish communities and fish information is very important. It's not in the past, but now it's still important, and that's why the research and also the information about the fish community, it's always interesting to be discussed. So now I open the question or also the discussion from the participant, and that's why fish is important, because we also eat fish. Okay, I see Safran Yustri raise hand. So Pak Safran.
Dear Dr. Ferse, my name is Safran Yusri from the Indonesia Coral Reef Foundation. During the development of your hypothesis, how did you select the current parameters, as there are a lot of parameters to determine improved ecosystem function? So it can be very frustrating for us selecting these parameters. Thank you very much, Dr. Ferse.
Yeah, that's a good question. So as Dea has said, there is always a lot of emphasis on fish, and it's usually used because it's an easy to monitor indicator, which tells you a lot about many different things. Yeah, so ecological processes, but also economic value, and the status of the reef. Is there overfishing? And they're strongly associated with the coral, and so on. And so for the coral, I chose 2 parameters. Was it 2? Because they're most informative for the question I was interested in, and that's survival rate. I didn't look at growth because it was too difficult to monitor, but it was survival and then number of recruits. And for the recruits, of course, species identity. And that was hypothesis driven. Yeah, I wanted to know what is the effect of the substrate for the transplantation, for the survival of the coral. So how are they doing in the long term? And then with regards to the coral, what is the effect on recruitment, so attraction or production? And for that, I also needed to know the identity of the coral. So that's why I try to identify the recruits. But of course, these are not the only things you can look at for coral. And if I would do a reef monitoring nowadays, yeah, so again, mind you, this was done 15 years ago, so the methods are also not the most recent. What I would look at more nowadays is in terms of species composition. Yeah, so what traits, what functional traits are present in a reef community? Coral cover alone, which is often still used in monitoring programs. We look at hard coral cover, but we can see a strong change in the coral community in terms of composition. What species are present? Do we see more, how are they called? Less branching corals, more massive corals, for example, because there is a selection against certain species from bleaching, from disease, and so on, and for other species. And so we can see a dramatic shift in the community composition and in the ecosystem services that are provided, like carbonate production, like fish biomass production, or coastal protection, or even tourism value. If we have more massive species, slow growing species, this might be very different, but we could have the same coral cover. Let's say 30%, 40% stays stable, but the composition, the species and the traits are very different. So nowadays, for monitoring purposes, I would always try to look at species identity and the traits of the species. What morphology do they have? How fast are they growing? Yeah, so these additional things. I hope that's answering your question.
Pak Safran, does answer your question?
Thank you very much, Dr. Forse. It's very enlightening. Thank you, Dea.
Okay, so another participant, Fauzan, maybe you also can tell where are you from.
Okay, thank you, Mbak Dea, for the chance. Good afternoon. My name is Fauzan Fanrianto. I am undergraduate student from Marine Science and Technology in IPB University. Okay, I would like to ask, I have 2 questions. First is, there's a lot of method of coral restoration, right? So in your opinion, which method is the most efficient for coral restoration? And the second, if I'm not mistaken, the natural restoration, it took 100 years, right, to recover. But if we did some restoration with human intervention, how fast the restoration with our intervention? Thank you.
Thanks for the questions. It's very difficult to say which one is the method, the best method. So I would always say the best method depends on your local condition, what you need to do. And I would say the best method is the one with the least, okay, of course it has to be cost effective, yeah. So the least disruptive to the system. If you do a coral transplantation, for example, it's a massive effort. Yeah, you have to take the coral from somewhere, you have to attach it. Usually that still involves diving. And so it's difficult to do at scale. You also still introduce a lot of foreign material, like artificial material, cable ties, plastic, glue, to the reef. And that in my view, should also be avoided. So try to use natural material as much as possible, and consider what are the local conditions, and what do you need to restore. As I've shown, and that is the case in many reefs in Indonesia, the natural processes are usually still quite good. There are, of course, some exceptions, like Teluk Jakarta or Kepulauan Seribu. If you look at some of the places there, recruitment may already be severely affected. Yeah, that you don't have sufficient recruitment anymore. But then also the local environment, the water quality is not good anymore. So you don't do anything in a reef with bad water quality, and where there's no recruitment and so on. Usually nothing will survive there. Yeah, you put in a structure, and what will grow on it is algae usually. But if you can improve the water condition, for example, yeah, that's passive management. Make sure there's less sedimentation, stop fishing in an area. These are passive, low-scale interventions, or you establish toilets on an island to make sure that human waste is not directly going into the reef. All of these are passive methods. They're not so sexy because they don't produce the images of transplantation, but they're much more cost effective usually in the long run. You improve the water quality and you support the natural recovery processes. And I would say in 80% of the cases in the Indonesian context, if you improve the environmental conditions and manage the reef better, it will recover. For those 10 or 20% where the substrate is in a bad condition, like rubble, and that's where I said it can take 100 years to recover because the substrate is moving, then you need to think of more drastic measures. For example, put concrete on the floor, or use natural stones. Yeah, this was done by Helen Fox and colleagues, and Trias knows this example quite well in Komodo National Park in some areas, and it was able to stabilize the substrate, and there was enough natural recruitment so that that the reef can recover. And only in a location where, for example, you have a very isolated island, there is no larvae supply from outside, and all the local larvae, all the local coral is killed, then there is a good case for transplantation because you don't have natural recruitment anymore. But Indonesia is a archipelago with lots of islands and a lot of currents. So there is larvae supply, I would say, to all of the different places in Indonesia still. So in my view, the need for coral transplantation is really, really small. There may be some examples where some transplantation is needed, but I would say most of the cases where you need restoration in Indonesia, management, passive restoration, or artificial reefs. And that covers 90 or 95% of the reef restoration cases, in my view.
Okay, Fauzan.
Yeah, that's a good answer. Thank you, sir, for the answer, and thank you, Madia, for giving a chance.
Okay, so any other question from the participant or discussion? Of course, Mbak Ade.
Thank you, Sebastian. Like I personally feel like we really need more science content for our marine and academic in Indonesia. And your presentation is very interesting. And I'm quite unwell actually these past 2 weeks, so I don't know whether I get your presentation right. And then someone sent me a message that your presentation is like one semester of lecture. You should split it into 6 sessions.
Okay, next time.
2 questions. Like you mentioned, like different numbers of recruits, right? Like the amount of recruits. Wait a second. Different amount of recruits on different setups.
Yep.
Yeah, that one. And I need you to explain that again, just like briefly, so if my question is right. Because you said more recruits in the setup with more structures and more coral transplantation, right? Is that correct?
You mean coral recruit or fish recruits?
Coral recruits.
Recruits, not recruit.
Coral recruits or fish recruits you mentioned.
No, I talked about fish, but these were mostly larger fish that are migrating. Not so much recruits.
Okay.
There may have been some recruitment, but in my view, it was mostly attraction, not production.
Okay, so you didn't calculate the coral recruits.
Yes, I did. So sorry, for corals, I looked at recruits, and for
Yeah.
fish, I just counted individuals. I did not look at, are they recruits or not. So recruitment really was for coral.
Okay, so for coral recruits, do you use tiles?
Yes, we use tiles. Okay, maybe let me share my screen again to show this.
I saw the picture. So you have the tiles in the 3 treatments, right?
Yes, exactly.
And then you find recruits higher in the area with structures and transplants?
Yep. So, okay, let me show this one again. So here, I hope you can see it.
Yeah.
There are six frames with plates on them.
Oh, yeah.
And so we had them on the 4 sides and 2 in the middle of the plots. And on each of these frames, we had 3 recruitment tiles. Yeah,
Okay.
and so 18 plates per treatment. And we removed them, then we put them in bleach, and we looked at the recruits. So here's what these frames look like. Yeah, so we removed the tiles, we exchanged them every 3 months, and then we looked at what kind of coral larvae, coral recruits are settling on them. And these are the results. Just to look at that again, and maybe I'll go into presentation mode because that makes it a little bit clearer. Which image are you seeing now? Is that the presentation mode, or is it you see the full screen?
Presentation mode. Full screen.
Okay.
Full screen.
Full screen.
So that,
Okay.
yeah.
You see my mouse now?
Yeah.
Okay. So control on the left side, middle is with coral transplant, and on the right side is with only structures. And you see that there really is no very big difference. Bunaken, in the 4th period, we even had the lowest number of recruits where there was coral transplant. Then in the last and the final measuring, we had the highest number where we had the coral transplant. So it's really this, the final measurements could indicate that there was some local spawning. Yeah. And that these brooded larvae were settling on the tiles. Yeah. But that's really only for one of the observation periods.
I see.
Maybe if we would have continued this, maybe it took 2 years for the coral to start spawning. Or to start release brooded larvae. If we would have continued this for another year, maybe we would have seen a different picture. But within the 2 years, we didn't really see anything. And also remember that most of the coral then died after 2 to 3 years. And so, maybe this approach is not suitable to really have an effect for coral recruitment, if that makes sense.
Yes. So my question with this is always, is the hypothesis where you have healthy corals, will it create a safe environment for coral larvae to settle? Do you think so? From your study, can you summarize that, or?
Well, I mean, I didn't look into the mechanisms behind this. It was only an observational study, and to do this properly, you need to do either, well, flume experiments. Danielle Dixon did some of this, but was heavily criticized for the way that she did the experiments. So maybe that's not an appropriate method. Or you use molecular tools, right? If we would have used molecular tools, we could show if the recruits were genetically identical to the transplants that we have. And that is still something that needs to be done, that you see like a genetic study of recruitment in the vicinity of transplantation to see if you really have an effect on recruitment. So at the moment, there are two hypotheses. One is that there is own production, and that's seeding the reef. And the other one, the second one, is chemical attraction. So just by having live, like chemical sensorial effect, I guess it's called, that by having live corals present, then coral larvae, they smell, this seems to be a good place, there's friends growing here, let's settle down. And that could be an alternative effect. And which one of these two is taking place, if there was an effect, I cannot answer.
Okay, and the transplantation, you use two different genus, right? Like the acroporids and the posiloporids.
Yeah.
And for the recruits, you don't like, do you also find these two genus or
families? We could only do it on the family level. So again,
Posiloporidae and acroporidae.
Yeah, Posilopora and Acropora. So these were the ones that I differentiated.
Mm-hmm.
Yeah, so the other ones were the rest, and Bunaken, the vast majority was, well, almost half towards the end was Posiloporids and some Acroporids. But really Posilopora, we didn't, on Bunaken, we did not transplant Posiloporids. Posiloporids were only at Meras and Ganga. Sorry, I didn't mention this. So on Bunaken, we really only had Acropora. And if you look at the black bar, yes, maybe this peak here, there is a difference in the final measurement, but for the other ones, there wasn't really any effect for the acroporids. So it doesn't seem that there was an effect of recruitment or transplantation, except for the final measurement period.
Okay, thank you, Sebastian. Clear enough. And the second question is a very quick question. Like you showed a quite small amount of CCA, and then if you do restoration or early life stage, they always said CCA is like a key for the coral larva to settle.
Yeah.
But I've heard like that's not really the case in Indonesia. I think the CCA is quite low. But we still have a high recluse or coral larvae settlement. Do you?
Yeah.
Like,
that's a good point. So we had the highest amount of CCA in the natural reef in Meras, even higher than Bunaken, even though the water quality in Bunaken was higher than in Meras. But so there was a lot of CCA, but not on the tiles. So the tiles, we replaced them every 3 months. And 3 months is not enough for CCA to establish. Yeah, so there it definitely did not make a difference, and we did not look at recruitment on the natural reef. There it could have been different, but my experience is really coral larvae will settle on almost anything. There is a lot of larvae. There is no lack of larvae, and we found them even on cable ties and so on. So they settle on many things, they don't grow on many things. The survival, of course, is different, but we even had some recruits on the bamboo substrate that we use at the bottom.
Okay, that's all from me. Thank you. I think we have question in the chat room there. Thanks, Bastian.
Yeah.
Sure. There's one from Nika, I see.
Another question from Nika from Leibniz Foundation. So
how far are the tiles from the natural reef? Yeah, that's of course a good question. So because for my PhD, I did not want to destroy any reef, so I had to use areas that were already degraded. Yeah, so where there was already rubble, and usually these plots were within a natural reef. At Bunaken, where we were working, it was mostly flat, also around it. So there were, I don't know, 10, 20, 30, 40 meters of rubble around it. In some cases, it was maybe 2 or 3 meters to the next patch of reef. So of course, it was not very far. Sometimes even immediately adjacent. Meras, it was like right in the middle of a reef. All my 3 treatments were in the middle of the reef, and you have normal reef, natural reef, right next to the treatments. And so the tiles, of course, also attract natural recruitment. Recruits, they are drifting for hundreds of kilometers, and most of the recruitment, or pretty much all of the recruitment, I would say, was coming from the natural reef, and very little, if anything, from the transplanted corals, if that answers your question. But in order to really answer how much was coming from the transplants, we would need molecular tools. So a genetic analysis of the transplants and a genetic analysis of the recruits. Then we can answer that better. But the distance was really not very far, a few meters.
Okay, another question. I think there is also a question in the chat. What is the maximum ocean space, occupation, or the coral reef habitat carrying capacity supporting sustainable coral reef? Do you think any coral introduction to any sandy or no reefs living in area before a good strategy for coral reef conservation? That's from Ivan.
Okay, that's a good question, but it's difficult to answer. I would personally say that the area, so the area of reef that we have in the ocean is less than 0.01% of the ocean is coral reef. Yeah? In terms of coastal habitat, usually most of the hard substrate, at least in the tropics, is potentially suitable for reef. Yeah? Only if we have sand or muddy area, then a reef is not establishing. But as soon as we have hard substrate, usually we find some coral community. And so I would personally say we have enough suitable habitat where there was coral before and that is now degraded, that we can focus on rehabilitating, that we don't need to create new reef areas in those plots where there was no reef before, like sandy area or so. Yeah, let's focus on those reefs where we had reefs before and better manage them and conserve them, than trying to create new reefs in those sandy areas. We have enough place to work with regards to hard bottom communities in the tropics. However, in some cases, like, okay, I see you're from Nusa Dua. In some cases, like tourism, for example, and that's an example from the Red Sea, it may make sense to create artificial environments, habitats, as, for example, a tourist attraction. Yeah, because we don't want the tourists to dive on the natural reef. They can damage the natural reef, and for divers, it may be enough to have some structure that they can look at, or that they can dive through, or where they can take selfies underwater. There it may make sense to use artificial dive parkours or statues or something. But that's not the same as trying to conserve a reef community. This is a very specific objective that we have for tourism creation and to reduce the stress on the natural reef, but it's not the same as trying to rehabilitate or to restore an ecosystem. These are two different things, if that makes sense.
Okay. I show another question in the chat. So that's from Made Merta. I guess this is from Bali, from the Southwest. And the question is, which one has a high survival, the coral from the transplantation or natural attachment coral?
Okay, since we're recording this and it's going to go on YouTube, I think I'm going to answer it in English there, and you can translate it into Bahasa.
Yeah.
Because otherwise I'm making a fool of myself, and maybe the meaning doesn't get across. But it's a good question. Terima kasih atas pertanyaannya. But it depends. There is no simple answer to this. It depends on the environmental condition. Yeah, a coral that is recruiting naturally in an area can have low survival if, for example, there's destructive fishing. If there is blast fishing or net fishing or so, then it may be bad. So the environmental conditions have to be good for the coral. The same for a transplanted coral. You can choose where to attach your transplant and choose a suitable area. So you have some power of selection. The coral, if it's recruiting naturally, it has to follow its senses, its instinct. Yeah, it smells the environment, and then it may think, okay, this is suitable, and maybe the water quality is good, but then there's still destructive fishing or cyanide or anything that can destroy it, or there's sedimentation and so on. So it really depends on what are the local conditions at the time of settlement, but also throughout the life of the coral. So if you want to increase the survival of natural corals, then you need to make sure that the environmental conditions are good, that there is no destructive fishing, that there is no pollution and runoff, and the same for the transplantation. I would say that a natural coral has the advantage that it already has dia sudah coba, ya, dia sudah merasa bagaimana kualitas air, so he's selecting a place to grow, and it's attaching itself. If you transplant a coral, you need to think, is this a suitable place, and can I attach it correctly? So it takes more work. But in the end, the survival of both of them depends on the environmental conditions. Pak Made Merta, does that answer your question?
Halo, Pak Made. Saya coba translatekan tadi jawaban dari Sebastian. Jadi jawabannya itu depends on atau tergantung dengan lingkungannya. Untuk lingkungan misalkan memiliki tekanan-tekanan seperti polusi, kemudian penangkapan yang merusak. Nah itu kemungkinan untuk penempelan akan sulit. Kemudian jika memang tempatnya itu sama misalkan, nah kita coba lakukan untuk alami dan juga tadi buatan, kemungkinan besar yang alami itu akan lebih mudah atau lebih survive karena lingkungannya tadi, atau karena yang transplant itu butuh penyesuaian dengan lingkungan, sehingga ketika sama-sama lingkungannya sama, kemungkinan besar yang alami itu lebih survivalnya lebih tinggi. Nah, tapi tadi untuk transplant sendiri itu memang kedua, baik transplant ataupun alami itu membutuhkan lingkungan yang cocok ataupun lingkungan yang baik untuk bisa tumbuh dengan baik. Seperti itu. Pertanyaan selanjutnya, is there any question or any sharing? I think we have another
Pak Abrar.
Oh ya, Pak Abrar. From your research, it was found that only a few species richness, namely Poeciloporidae and Acroporidae. In your opinion, based on experience, how long does the research take to get a more complete community from the coral recruitment process at the restoration site? I think it's important to predict how long it will take for natural recovery to occur, because usually from transplant, its development is limited. So, yeah.
Yeah. Okay. Of course, the community that we had in our experimental plots was not a natural community. We selected these species specifically because of their reproduction modes and because of their growth modes. Yeah, so they're easy to transplant, and some of them were brooding and some of them were spawning. So that's why we selected them. But it's not a natural community, and that's also important to keep in mind when looking at the fish community. Why maybe we would have had a different response of the fish community if we would have used more coral species. Yeah, so that's important to keep in mind. And a natural community to reestablish, assuming that everything else is okay. Yeah, if we have good environmental conditions, if there is no climate change, which is idealistic, but under ideal conditions for a natural community to reestablish, including slow growing species, massive species, will take several decades. You can see very fast recovery just in terms of coral cover. For example, after the bleaching in the Maldives in 1998, we lost 90% of the coral cover. And then if you look at coral cover just a few years after, it was almost the same as before, but it was mostly Acropora species that were regrowing, not a diverse coral community. And until you get a diverse community to establish, this will take several decades, and it's not something that you will observe within a short experimental duration. Hope that answers the question.
Fabrar? Is it enough, Pak?
Okay, thank you, Sebastian. Hi, I'm Abral. I hope you still remember me in the scientific diving training in Bunakan last year ago. Okay, yeah, I mean, we know that the coral recruitment coming from Acroporidae and then Fossiloporidae is annually spawning. It's every month spawns. But as we need how long another recruitment come from another species. I think it's more than years, maybe 2 years, 3 years, and more again maybe.
Yeah. Yeah, thanks, Barbara. The natural recruitment is, of course, it's a critical factor because it's very variable. It may be different from year to year. We don't have the same composition of recruits every year. And some species, the brooding ones, they produce continuously. Yeah. So for the brooding species, they're usually less, but more continuous. So that will probably within a year or two or 3, you will get most of the brooding species that are available. But for other species, yeah, it's very variable, and the rarer species is the longer it probably takes until it shows up and survives and recruits and establishes itself. Yeah, we have several bottlenecks. We need the larvae, then the larvae need to recruit or need to settle, and then they need to survive until they're adult or big enough that they survive this bottleneck. And so these 3 factors together mean that there is a long process before we get all of the different species established again. And that by itself takes a long time. But this natural succession that we have in the community. Usually we get those fast growing, mass spawning Acropora species, Seriatopora, and so on first. And then eventually over time, we get small individuals of the other slow growing rare species. And until they can establish themselves, that's why it takes, 20, 30, 40 years for a natural community to establish.
Okay, thank you. Just info that I have already done to the coral recruitment monitoring in the Indonesian coral reef gardens is last year. It's also the same in the 1 year. In the 1 year, just only found the Acropora and Fussulpora also. I think it's the same. Yeah, the same. Thank you, Sebastian.
Yeah, thanks for the question, Pak.
Yeah.
Thank you, Pak Abrar. Another question from Bahari Prawara MPA. Is there any possibility the coral grow faster after they got bleaching?
To my knowledge, when corals recover from bleaching, usually the growth is reduced because they need to use a lot of their energy to recover. So other processes like growth or reproduction are reduced. They choose to use the energy for survival and not so much for growth. So normally the first response after bleaching is a reduction in growth. And you also see that when you look at the growth rings. So when you cut the coral, it's like trees. You can see rings how they grow. And bleaching you can usually see because it's a period of slow growth. So less growth, not more after bleaching.
Okay, we have another question from Gayatri, please.
Thank you. Hi, Sebastian.
Hello.
Hello. So I'm just sharing what we are doing in North Bali. Before Pak Abrar already mentioned that he did have a tiles in the sites where the Indonesia Coral Reef Garden was conducted in 2020. So basically, I'm talking about the same area. And my colleague at LINI, they conducted the monitoring of the natural recruitment on the artificial structures. And so this is the 3rd year. And the data from the 3 years, I think over 20 genera of the stony corals were recorded. This is juveniles, so it's not it's not recruits, so it's already juveniles. And what we learned that also this the majority, the dominance juveniles probably likely came from the nearby natural reefs, because we find the same species. Of course, DNA study need to be done, but without studying the DNA, we can make an assumptions, I guess, because there are a big colonies of the same genera. What is interesting is that we also learn, in the last few days from Professor Suarsono, that the natural recruitment needed help from us, because we put the artificial structures in there, so it's a human intervention, but we also need help to what you call it, for the other organisms like sponges and the algae, that they also attached to these artificial structures. So the artificial structures is from the concrete. So then in order to help this natural recruitment to grow, as it still need the human intervention, like cleaning the sponges surrounding the natural recruitment. Otherwise, they will compete it, with all these sponges and green algae and other creatures, tunicates like that. So I know that it is true that takes decades for the reef to reach the equilibrium of the function of the reef. However, there are effort of this natural recruitment from the surrounding colony, from the natural reef. So we think that the factors that influencing the natural recruitment on the artificial structures, they are variable, but it's also without intervention from human, then it will slow the growth of this natural recruitment that already survive, like the 30 years, but would probably die because of the competitions from other organisms that attach to the natural reef. That's just the sharing. Thank you.
Yeah.
So my colleague in the village in Les, their internet not stable, so I'm just passing on the message from what they're studying. Thank you.
Thanks, Bu Gayatri. It's very nice to hear from Bali. I was fortunate enough to visit Desales, was it 10 years ago, I think. So I'm very impressed by the work that's being done there, but I haven't had the opportunity since then. And I agree that probably a lot of the recruitment that takes place is from local sources. I think even though there is this potential of larvae to drift very far, maybe that was overemphasized in the studies in the past, like 40 years ago or so. There was a lot of dispersal capacity of corals, hundreds of kilometers and so on. I think that's possible, but in my view, the vast majority of recruitment is taking place in a vicinity of a few hundred meters to a few kilometers. Yeah, so the most of the corals will be locally sourced. Also, if we look at the community composition, and I didn't show the results for the natural structures or the concrete structures. I also looked at one snapshot at the recruits on my structures, not just on the plates, and there the community composition was quite different. So what you see on settlement tiles is usually dominated by pocilloporids, acroporids, with a different spawning strategy. You have lots of recruits, but very many of them die and don't survive until juvenile stage or later. And so if you look at the established community on concrete structures or anything else, it will look different from the ones on recruitment tiles. And it usually looks more like the natural community composition. So my impression is the same that you've just shared. And also with regards to the need to maintain and sort of support the recruits or the juveniles, I partly agree with that. I think we can do it, and we can probably increase the survival. And maybe nowadays, because the condition is critical in many areas, that may be needed. But I would say under natural conditions, if the local conditions are good, then natural processes are sufficient for a community to establish itself. But I agree with you that it takes a very long time, and oftentimes we have different stressors like agricultural runoff, water quality is bad. There is some overfishing, even in community managed areas. We have less fish than we have in a completely undisturbed reef, and so that means that herbivory is also reduced. And then it certainly is helpful if we invest some energy and clean and really maintain the juveniles, and then they can survive better. So thanks for sharing those impressions.
Yeah, thanks for the sharing, Bu Gayatri from Bali. We still have another question and discussion, Sebastian. there is a question from Nurul. She wants to ask about the coral restoration. When we want to do a coral restoration, do we have to adjust the coral family that lives in the area, or we can choose a coral family that grow faster or survive in that area? So this is how to choose the species or maybe the family for the coral restoration.
Yeah, that's a good question, and it goes a little bit into the area of assisted evolution or assisted migration that is discussed in reef restoration and reef science. I would always be in favor of first using local species and the ones that have grown or are growing in the area, because there's a reason why they're growing there. If other corals are not growing there, then we need to understand why they're not growing there. Is it because of limited connectivity or limited availability, which I doubt in the Indonesian context, because there is a lot of exchange and there is a lot of connectivity, although we do have a marine Wallace line and so on, but there is a lot of exchange possible. If we choose another species for some reason that it grows better, we need to make sure that it is really growing better in those local conditions. And if we're introducing a species, so if it's one that's growing on a neighboring island or so, then I would always ask myself, why is it not growing in your location? There must be a reason for it. If you're looking for species, and that's what this assisted migration, assisted evolution is going into, to take species from, for example, the Arabian Gulf or the Red Sea, where they're used to different thermal conditions, maybe are more bleaching resistant, then it really introduces the question of introduced species into a new system where it wasn't before. And that at some point is dangerous because you change the local species pool and the local gene pool. So with really bringing in species from the outside that do not exist in an area, so outside of the natural distribution, I would be very careful. If it's bringing them in from another site, yes, maybe there's a reason, selecting them for faster growth or higher resilience, but ask yourself, why is it not growing there in the first place? And so will it really grow better as I expect it to if I bring it in from somewhere else? So that's why my recommendation would always be work with the species that are already present at a site.
Okay, that's enough, Nurul. Yeah, so I'm so happy because of a very interactive discussion and also the spirit of the participant in this score session too, because many questions, many sharing, and I think we still have another question from Pa Martin. Do you have some reference or guidance on site that indicate highest of coral recruitment level based on your study and experiences? Which site that's suitable for requirement to deploy the coral reef restoration structure from CTC?
Based on... Hello, Pat Martin. Thanks for asking the question.
Yeah, hi, Sebastian.
That's a good question. So yes, I would always recommend to do a recruitment study first, if we have unlimited time and resources. Of course, I know that's not always possible, but to my knowledge, we still don't have sufficient understanding, because there's not a whole lot of collection of data on recruitment. Recruitment is very variable, so I don't think we have like a map that's showing recruitment potential throughout Indonesia, or that's showing the different current systems and where the corals are, the larvae are accumulating. I think it would be possible, but it takes a lot of resources. And so I'm afraid there is no easy answer to that, which sites are very suitable. So the best we can do is, if we have the time before we start a restoration to invest some resources in looking at recruitment first, like for a year, for example, ideally, and then seeing, okay, we look at 10 different sites. These ones have the highest recruitment potential, and then maybe we put our artificial structures there. But I know oftentimes we are limited by resources. We have short project cycles, so that's not always possible. But ideally, that's what I would recommend. Does it help a little bit?
Oh, Pak Martin. Yeah. So
Yes. Thank you, Sebastian, for your answer. Yeah.
We still have 9 minutes for this session. I think we have another question from Muhammad Khalil. He is from the Fisheries Diving Club IPB, and the question is, based on your research and experience, is there another coral disease expect bleaching that intervenes coral recruitment growth, either on ER or the nursery?
Yeah. Okay, disease in a wider sense. I think bleaching is one of the biggest threats. So if you have, a heat wave, then it's affecting all of your transplant nursery sites and so on. In my experience, corallivores are the next big thing. So crown of thorns, Grupella, or even some small, what are they, like small flatworms. I had coral parasites that were actually very small nudibranchs when I was doing my work in Karimun, Java, and they were feeding on the Pavona species that I was working with. So a lot of the Pavona got infected and eaten by tiny nudibranchs. So that can happen in my, but I don't have data on it. Yeah, it's more experience and what I hear from other people. If you have transplants, because you're wounding the coral, you're injuring it, and maybe that releases some odor that attracts corallivores. So what I'm seeing on coral farms, for example, and in nurseries, is that you tend to get, if you're unlucky, a lot of corallivores, more than disease. I think other disease like white spot disease or black band disease and so on is the likelihood is very small. So heat wave and corallivores.
Okay, thank you. And maybe before we close the session, I would like to invite Pak Begin. Hello, Bang Begin. Hello, I show you there. Mbak Ade?
Ya, Gin.
You have your closing statement.
Nggak bisa denger nggak? Om Begin. Nggak denger apa? Ntar. Nggak. Nggak bisa jalan kali ya itunya, speakernya. Nggak apa-apa, saya aja yang ini, yang Pak Begin. Nggak bisa ya, udah ya, kira-kira saya aja yang tutup.
Ya.
Terima kasih banyak, sangat menarik ya Sebastian. As you can see, there's a lot of interest, there's a lot of curiosities, and a lot of questions beyond what you have presented. So thank you for coming. This is really valuable for us at SCORS and also in general for the coral reef science in Indonesia. We hope like we can in the future work, we can integrate more science into our restoration work, and where we can publish the work, and then the information shared can be used to improve the restoration practice in Indonesia. No, your bahasa is all good. Don't say sorry. Thank you very much. And we hope to see you maybe in person in Indonesia in the near future. And also for anyone who are interested to present their work on restoration, reef restoration in Indonesia, you can contact me or Pak Bigin through emails or chat. And also we will meet in 2 weeks' time, and we will have Arnold Rival from Yayasan Orang Laut Papua to share his work in Raja Ampat in 2 weeks' time on Monday. And anything else? Do you want to say something to close your presentation, Sebastian? Or that's all?
Yeah. Sure. Thank you again very much for the invitation. It was my honor to present in front of the Indonesian reef restoration community, and I think this exchange is really important. I'm also honored by the very high number of attendants.
Yeah.
And, the different people that ask questions. Pak Abra, Pak Martin, Bu Gayatri, thank you a lot. Yeah, and let's stay in touch. I think it's really important that we have more publications also, even if it's, you know, a small report, anything, but that is published from the experience from reef restoration. That's like my take-home message, I hope. Even if it's failure, yeah, I mean, many of the things that we had tried in the study did not really work out, but it's also important to say, you know, this substrate does not work. There is limited evidence for recruitment at this time scale. Maybe it's not the most useful. We did not see such a strong response to coral transplantation. Yeah, these are all things that went different from what we had expected, but I think it's important to share that experience, because otherwise everybody keeps repeating the same things and we don't learn from each other. And this exchange is really important. So thank you.
Terima kasih banyak Sebastian for the important message, the take-home message, and then let's work together, everyone here. Also Sebastian for the international support, and I only wish sorry, that we will improve the reef restoration practice in Indonesia to the near future. And please, yeah, Pak Veda Santiaji also, who supported us with the WWF CTI, say thank you. Sorry, and I think I should stop talking. Thank you so much, Dea. You can take over.
Thank you, Mbak Ade and Sebastian. Like what Mbak Ade said, that 2 weeks later, we will have another score session. So you can stay tuned in our Instagram to get the information and also the link for the meeting or for the Zoom meeting. And thank you very much for the participant. Like I said before, I'm so happy for the enthusiasm and also the spirit of the participant who attend this afternoon. So see you again in the next course. Hope everyone stay healthy. And thank you very much, Sebastian.
Thank you, Dea. Thank you for moderating.
Yeah.
Thank you. Bye-bye.
Bye.
Bye.
Thank you very much.